All right, I think it's about that time. How do you guys like this weather?
No.
No, I got some no's. Any yeses?
If there's no, it's fine. It's really fine.
So I was in New Jersey this weekend.
That's where I'm from.
What?
I'm from New Jersey.
You're from New Jersey, yeah. So we were up in southern, near Flemington, Allentown. My in-laws live there. And I had to sit there warming the car up with my foot in the clutch because I couldn't even move the gear shift lever. No thank you. A couple of reminders. We do have a discussion session on Friday. This is the problem set. Your product from this discussion session is just going to be you turning in the problem sets that I posted here. I'm not going to grade them. You turn them in, you get a 10. Simple as that. The point of this is to make sure that you give it a try and see where your strengths and weaknesses are, come up with questions. On that discussion session, I'm going to stand up here, I'm going to say, which problem do you want me to solve? And we'll go through whichever ones you're interested in until the time is up. If you wish instead to go through some of the old exams with these problems, that's fine. Whatever you want to do. The following Monday, the 25th, I will hand out the take-home section of the first midterm exam. That exam is coming right up. February 1st. We'll do the Yellowstone discussion session before that, and then we'll jump into all the bacteria. I also have a plea for some help. I'm interested in any interesting papers that you guys have run across of microbiological significance recently. Something cool, a very recent paper that you'd like to talk about in the discussion sessions. We did the devil facial disease the last time. I've got this one to talk about the Black Death. I kind of like to synchronize these with the lectures a little bit. So for example, we're going to talk about thermophiles and that kind of stuff. It's really this discussion session up here that goes with that. But if you've seen a paper recently about life at high temperatures in some way, or extraterrestrial, something like that, that'd be a great one to talk about. Likewise, photosynthesis, although this one will probably do the Black Death here after we talk about the proteobacteria, since Yersinia is a gamma proteome, and so forth. So, you know, if you've run across something in class, if you've run across something in any way I mean, cancer's facial disease in Tasmanian devils isn't really that microbiological, so I'm pretty flexible about that. Whatever you want to talk about. Yeah?
So, theoretically, if classes cancel on Friday, should we...
We'll still do the discussion session.
But should we, like, send them... How are we going to turn the next week?
Yeah. Yeah. Just take a picture with your phone and send it to them. No problem. Yeah, I'm supposed to go down to Fayetteville after class on Friday for a North Carolina Academy of Science meeting. I don't know whether that's going to happen or not. All right. So we've talked about, let me go ahead and pick this up here. We've talked about basic tree construction using the neighbor joining method, which is a least squares distance matrix approach. And it's pretty simple, right? I mean, you sit there with the sequences, you align them, which you know how to do. You create a distance matrix, you convert that into you create a similarity matrix, and you turn that into a distance matrix, and you do a neighbor joining tree, and you're done. Pretty straightforward. If only it were so simple in real life, with real sequences and real circumstances. There are any number of complications to this, and complexities, and phylogenetic analysis is an entire field of study in kind of the genetics quantitative biology area. So we're going to talk about some of those complexities because they do make a difference. And we're going to start with alternative substitution models. So when we talked previously about calculating similarity from two sequences, we just took those two sequences and at every position along the alignment, they were either the same or different, right? It's entirely digital, plus or minus. Is it the same or not? But of course, it's more complicated than that, because if you've got two bases, let me give you an example. If there's a marker anywhere. So if you have two sequences, three sequences, and at a particular position, one has a G, one has an A, and one has a C. These, of course, are all different, right? And when you're comparing them pairwise, they will always be different. But are some bases more different than others? Sure they are. These two are purines, right? G and A. This one's a pyrimidine. Oughtn't this difference count as less of a difference than this difference? Pragmatically, if you look at sequence alignments, you see that these purine to pyrimidine differences are a lot less common than purine to purine or pyrimidine to pyrimidine. These, of course, are transition mutations, and these are transversion mutations. You don't know which came first, of course. They're just differences. Transitions are much, much more frequent than transversions, and so ought to count as less of a penalty, as less of a difference. And so this is the Kimura two-parameter model, where instead of it just being binary, same or different, it's same, transition, or transversion. There are three options. Another way to look at this is if you've got GA, C, and U, a transition mutation, G to A or C to U, might count as half a difference, whereas a transversion would count as a full difference. And you can adjust that parameter, right? I mean, the transversion is always going to be a full difference, but what about the transition? And usually the way this is done is not just taking a number out of the hat like 0.5 difference or something like that, but you take your alignment and you do a pre-sifting through that alignment and count the number of transitions and transversions. And then you set the transversion parameter to complete difference, but the transition mutation counts based on its relative frequency to transversions. So if transitions happen 25% if transversions count 25% as often as transitions, then the transition would only count as 0.25 difference. And so you calculate that parameter right at the onset as part of the analysis of the alignment. Well, you can go one up from that, right? If you've got GA, C, and U, there are actually six kinds of differences. It can be a G against an A G against a U, a G against a C against an A C against a U, an A against a C, U, an A and U, etc., right? 6 possible juxtapositions. So why not calculate a difference parameter for each one of these? And so you go through your alignment and you sift through the relative frequencies of G to C, G to A, G to U, and so forth, calculate or determine the relative frequencies, and use those numbers to have a parameter, a penalty when you see a mismatch like that. Now, in order for this to be very helpful, your alignment has to be large, right? You've got to have lots and lots of transitions and transversions to look at. And for many alignments, the data isn't sufficient for this to be very helpful. And so these 6-parameter models are less commonly used than the 2-parameter models. There's another really cool way to determine these weights, and this is not mutually exclusive with the others. You can do both of them. And this is called a weighted analysis, position weighting. And so here's a chunk of an alignment from ribosomal RNA. And if you look at this, you can see that there are some places that are very highly conserved. And I apologize for anyone who might be color deficient. Color makes an incredibly big difference in how easy it is to look at alignments, at least for me. And so, for example, this position right here is almost always a U. Here's an N at that position, meaning they couldn't determine the base. So that's a very highly conserved position, right? Shouldn't a change in that position count as a big penalty? If it's very, very highly conserved, then an organism that has a different base there, that ought to count for a big difference. Whereas other positions, are highly variable. They can be anything. And so a difference there ought to be a relatively minor penalty. So what you can do is take your sequence alignment, and for every column in that alignment, calculate a variability number. How variable is it? Now you can do something really simplistic, like say it's 50% conserved. That's not usually very useful because you don't know what the other 50% is, how the variability is. Normally you would use an entropy coefficient that I won't get into. It's an information theory thing. But you can calculate numerically what the variability of a position is. And then use that, multiply that by the difference to count as a score for penalty. And again, you can also do the transition versus transversion, a 2 or a 6 parameter model on top of that penalty. And so multiply the penalties against each other to give you a relative weight. I point out this weighted neighbor joining method because those of you in lab, when you do your term project online at the Ribosomal Database Project, you'll see the analysis called WABER, which is a weighted neighbor joining approach. And so with big alignments like are available for ribosomal RNAs, this is a really powerful technique and helps the process a lot. For smaller alignments, the positional variation isn't as useful. Now here's a really important one though, and this is a complication that doesn't get enough interest. There actually aren't 4 possible things at every position in alignment, right? There are actually 5. It can be G, it can be A, it can be C, or it can be gap. And so the question is, what do you do with gap? If you've got one sequence with a base, like this C, and another position with a gap, how do you deal with that? And how sophisticated do you want to be when you deal with that? A really simplistic approach is to consider gap just like any other character, right? And it's either the same or different. C to gap, gap to gap. These are the same, these are different. Done. You can include it in it wouldn't be a 6 parameter model, it would be what? A 10 parameter model, I think. That's not the way most of these programs do it. Most of the time, when a training program sees a gap, it ignores that position completely. It won't calculate it, it won't count it either as the same or different. And so if you have an alignment of 100 positions and you have a gap, then you take however similar they are divided by 99 instead of 100. So you ignore the gap. Now why do people do that? There are a couple of reasons for that. Notice these sequences here that have got all this empty space. These sequences here. Those aren't really empty spaces. What's present here is the data that's available, not the entire ribosomal sequences. These are partial sequences. They were obtained by PCR amplification, probably. And as a result, you only have a chunk of the sequence, not the whole thing. But when you put that in alignment, you put gaps in to align up all the stuff. What this means is that when you see a gap in alignment, you don't know whether that gap is the absence of a nucleotide or if it's absence of data. I've lobbied for years amongst people doing sequence alignment programs to have two different kinds of gaps for this reason. A gap that represents no information, maybe a period, and a gap that represents the absence of a nucleotide, which is a real gap. But since programs don't incorporate that, generally speaking, you don't want to count all of these as differences, or these sequences will tree out way off in I don't know, limbo somewhere compared to the rest of the sequences. It will create a huge artifact in the trees. And so in order to compensate for that, for example, when comparing this sequence to this one, you just ignore all this stuff and only count where you have nucleotides for both of them. There's another reason why gaps can be ignored, or are often ignored. And it has to do with the fact that you typically, when you're counting similarity, you do it one column at a time. And usually, if you're counting gaps, a gap is a big penalty, right? Because the insertion or deletion is going to be much rarer than a nucleotide substitution. So it ought to count for a big difference. And so for here, for example, let's see. This one has an A at that position, and this one has nothing. And so that's a single difference, right? It's a very big difference. But what about this one? It's got a gap here too, but it's also got a gap here. Now that's two big penalties against that sequence compared to this one. But are those gaps really independent? Probably not. Probably this represents not two individual insertions or deletions, but one insertion or deletion of two nucleotides. This is a big difference when you're looking at something like this, right? Because here you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 nucleotides that are gone and present in something else. Do you want to count that as 16 big differences, as 16 insertions or deletions? Or do you want to count it as one insertion or deletion of 16 nucleotides? That's probably the best way, right? And so some more sophisticated treeing algorithms, when they see a gap, will actually combine those, any adjacent gaps, and will have a gap opening penalty, which is pretty large, and then a much smaller penalty as the gap gets larger and larger. Okay, please stop. So we talked about ratios of transitions and transversions and how deletions and insertions work. Doesn't it sound like we're describing how RNAs evolve? This isn't just a numerical comparison of two strings of letters. This is an evolutionary model. You're trying, in the mathematics of scoring similarity, you're trying to capture how RNAs evolve. And so, while some people call these substitution models, what you're really creating is an evolutionary model. You're trying to score how much difference some amount of evolutionary change is. There's one really interesting case that you have to deal with sometimes. Someone asked earlier about sequence convergence. This does happen. Not very often, and it's something you can deal with, but it does happen. And the biggest one has to do with GC bias. If you look up the genomes of some organisms, you might see that some of them are 60% G plus C, and some of them are 30% G plus C, and everything in between. In fact, they go a little wider than that. Organisms have, because of their nucleotide metabolism, preferences for GC or AT base pairs in their DNA. Let me give you a really simple example of this. We may later talk about SAR11, this tiny, tiny organism that lives in the ocean. and is specialized in eking a living out of the tiniest levels of nutrients. Nitrogen is precious to this organism. It can't make its own, it can't fix its own nitrogen, and it doesn't have a lot available. And so it does everything it can to scrimp, to recycle, and to minimize the amount of nitrogen it needs. AT pairs have less nitrogen in them than GC pairs. And as a result, this organism has a GC bias in its genome skewed well to the AT richness instead of GC richness. Other organisms, sometimes for reasons that are unclear, sometimes for reasons of life living at higher temperatures, GC pairs are stronger, right, than AT pairs. And so in the DNA, the melting point of the DNA will be higher if it's GC rich than if it's AT rich. And so thermophiles, under certain circumstances, tend to have genomes that are richer in GC pairs. And so you might be creating a tree that on one hand has a thermophile in it, and another hand has, for example, Rhodospirillum rubrum, which for reasons are unclear is GC rich. And that GC bias will skew the tree. It will tend to push those sequences together artificially, because wherever a base flips back and forth between A's and AUs and GCs, in the thermophiles they're going to be GCs, and in Rhodospirillum they're going to be GCs, and so you have this kind of convergence. There's an easy way to correct for this. It's not a great correction, but it's a useful one. And that is to take your sequence alignment and make it, instead of A, G, C, and U, make it R and Y. R for purine, Y for pyrimidine. And so now you don't distinguish between G and A or between C and U. You're only looking at purines versus pyrimidines. Now, pragmatically or technically, the easiest way to do that is to do a global search and replace every A with a G and every U with a C. It's just a dumb trick that you can use. And then do the trees. Now you've thrown away most of your information when you do this, right? Because most substitutions are transitions. And you've erased all the transitions in the alignment. But the data that remains is free of this G plus C bias. And so it's a great way, if you've got a tree and you've got a question or concern about convergence, then you can run one of these GC trees and make sure that what you're looking at in the tree isn't an artifact of this GC richness. Normally you don't publish those trees, but you mention that you've done it, and it confirms that it's what you think it is. Here's the big one, though. Long branch attraction. All phylogenetic tree algorithms are subject to the danger of long branch attraction. But it's really easy to see where this crops up in neighbor joining. And so let's imagine that the real relationship between three sequences here is A, B, and C. This is the actual natural relationship between these organisms. What's going to happen when you do a neighbor joining of this tree? This distance is going to be short, right, compared to either this to this or this to this. This will be, at some point, this will be the shortest distance in the tree. And it will join those together as part of the algorithm. That's what neighbor joining does, right? And then it will try to adjust the branch lengths to fit as best it can. It will not be a great average, but it's forced to do so. And the result will be a tree in which A and B are specifically related instead of B and C. This is called long branch attraction because if you have a tree with short and long branches together, the long branches will tend to cluster together. Of course, the flip side of that is the short branches also tend to cluster together separately, which is what is happening right here. So you could call it short branch attraction, but it's also long branch attraction. And what this means is anytime you do a tree and you see branches that are significantly different from each other, you need to start worrying about this artifact. and whether what you're seeing isn't what you think it is. Very often the trick would be to add some more sequences related to this, and then pull sequences out and add them into the tree and see what happens to it. We're going to talk a lot about long branch attraction because it creates some interesting questions in phylogenetic analysis.